Well, welcome again then to the first school, and thanks to Eileen and to Gerhard for involving me in it. When I accepted Gerhard's invitation to speak here, I did not know at that point that the first school was in fact the first information theory school in the sequence of information theory schools that my late colleague Sergio Cervetto had been the prime mover of pushing through the IEEE Information Theory Society, and appropriately, of course, this first school has been dedicated to him by its organizers, so thank you for that. Sergio and I were quite close. He was an assistant professor at Cornell for the last three or so years out of the 37 years that I was at Cornell before moving to Virginia. So when he would come down and visit, and so this is nice for me to be able to participate in this event for that reason as well. As you know from the program of the meeting, I'm going to be talking to you about a totally different kind of topic that is not directly connected to the others, in the sense that it is not about man-made communication systems, but it's rather about an aspect of information theory in life sciences, but it's particularly some connections between neuroscience and information theory, for that matter, neuroscience and I guess what we would call system science within the electrical engineering curricula generally, not just information theory, but other things too, like signal processing and random processes and all of that. My particular approach, of course, especially in this lecture and to some degree in the one that I'll give after it, is to connect to information theory, but you can use a lot of the other things that you've learned in electrical engineering experiences, undergrads, grad students, postdocs, whatever, to attack some of these problems in neuroscience. The great difficulty, far greater than it is for you to make progress in your research, it's the same difficulty, but it's just at a whole higher level. The difficulty always is, what's the problem? How do I formulate the problem? And this is true of all research. But when you try to get into this particular interdisciplinary area where you need to understand another science that you didn't grow up with, and then get to the point where you can ask the right questions, and I'm not sure that I'm there yet. I was certainly not fully there at the time that I gave the original Shannon lecture, and I'd already been at it for 5 years. It's difficult for a variety of reasons. Most biologists, including most neurobiologists, neuroscientists, maybe it's too strong to say don't like mathematics. Maybe what's worse is that they don't trust mathematics. And they don't trust theory at all. And they have a good reason for not trusting theory, because it's been ripped out from beneath the underpinnings of biology several times over the centuries. Most recently, of course, in the 1950s, when the whole DNA approach to what it means for things to be alive and how all the living information is put together was completely different than everything that had been taught to people for several hundred years before that. And therefore, if you are, as most of these people are, neuroscientific experimenters, then you not only don't, you say you go out of your way not to tie your experiment to any theory. You say, when you look at this vast bunch of literature, and what you see is descriptions of experiment after experiment after experiment. They say, here are the hypotheses, here is the method, here are the controls, here are the results. Some discussion, maybe sometimes as a hint in the discussion of why it is that they looked at this, that it fit into some big picture. But they would prefer not to tell you that, because when that big theory thing goes down like it's done before, and they tie their experiment to it, their experiment will die. And they feel that their experiment is valid in itself, has a life that isn't connected to the theory, and therefore when the right theory comes along, if this experiment was done right, of course it has to be relevant, appropriate. So they don't. Then on top of that, as information theorists, I can tell you this, that I have a complete, I think it's by now complete, copy of all the information theory transactions in hard copy. And it sits, some of it at home and some of it in the office. If it were all in one place, it would be on a shelf about this high, and it fit between here and the end of the wall. When I go into my neuroscientist's office, my collaborator, he has a long entryway into the office, which is at least half the length of the front of the room and higher. And on that shelf are some of the neuroscience journals of the last 4 or 5 years. When you go to Neuroscience 2004, which is the last one I went to, I'm going to get this now. Okay. There are 30,000 participants. There are 15,000 papers that are presented, as many as all of information theory from the beginning in one meeting. How do you present 15,000 papers in one meeting? And I mean, God, how do you know how to get to them? Well, there are 5 books, Monday, Tuesday, Wednesday, Thursday, Friday. There's a CD. You take the CD, you plug in what your topic is interested in, and it will tell you where on this giant convention floor to go between 9 o'clock and 12 o'clock to this poster and that poster that are relevant to your thing. So it points out where they are. You can put that together if you can stand it for more than I couldn't take it for more than a day myself. But between 9 and 12, 1,500 posters are put up. Between 12 and 1, they're taken down, and 1,500 more are put up for the afternoon. That's 3,000 a day, 5 days, 15,000 poster papers. There are also some other presentations. You know, this is one meeting. You know. Now, 98% of it you don't give a darn about. In fact, I suppose 98% of it, any given person there probably doesn't give a darn about. But to find out what the other 2% is terribly difficult, especially because they don't want to tell you necessarily in the way in which they write it. And there's really only one way to do this, and that's, of course, to hook up your research with somebody who's been doing that all of his or her life, and that's what I've done. So if I can make this thing advance, which just doesn't seem to be easy to do. So click on it, doesn't advance? Okay, it did. Now I got it to advance 2 times, and that isn't what I wanted either. This is a complicated little mechanism, but nah, it just doesn't go backwards the way I want it to, so it's a big problem. Okay, so I go to previous, like that, and I click on previous.
If you want, it might be easiest just to use the keyboard.
I really don't like it like that. Can I just push them like that? Yeah, I'm going to do it that way. Okay, so I have bio collaborators. And the principal one of these bio collaborators is Chip Levy in the University of Virginia Medical School, whom I started doing this collaboration with long before I went to Virginia. It goes back to 1997, really. You may think that that's why I went to Virginia, but that's not why I went to Virginia. I went to Virginia because my daughter's family is there and all 4 of my grandchildren, so that's why I went to Virginia. But this certainly was a secondary motivation. Jim Mandel is also on the medical faculty there at UVA, but Jim happens to be the father of those 4 children, so that's. But he and I have written some things together too. He's a neuropathologist, and okay. But it's principally Chip that I work with. So, and this talk is, as the first slide said, a revisit or reliving of the Shannon lecture that I was privileged to give in 2002 at Lausanne, which I called Living Information Theory. So this is Living Information Theory relived, about 5 or 6 years after, to come back and change a few things and omit some things and add some things. Especially, I hope, if I get far enough, to add the proofs, which of course I left out of the Shannon lecture because of the nature of the time that's available and the background of spouses and other sorts of people who sit in the audience at the Shannon lecture, plus the fact that they take 15 minutes introducing the whole thing. But, you know, so this is okay. But the object here is to model, and I should say that why I called it living information theory. It was meant to be, and is still meant to be, a triple entendre. One, of course, is that it's talking about information theory in living systems. Another is that I've been living information theory for all of my research life. So, and the third was that I wanted to help dispel the occasional rumor that information theory is dead or dying. So, you know, I don't think that information theory is at all dying. And this is one of the areas in which my personal belief is that in this century and probably beyond, information theory will have some of its impacts, I think, because life sciences seem to be, probably more than anything else, well, it's been said by certain pundits that the 1900s were the era of the century of physics, and the 2000s, that's the century of life sciences. So if your discipline does tie to the life sciences, and you're so inclined, then maybe you should think about that. I guess I had some things in my rate distortion book, even back in 1971, that little section in the back that tried to say a little bit about connections on a statistical mechanical level between information theory and try to tie that to living systems. But that isn't anything I pursued for the next 30 years, really. I came around to it more recently when I got to be of the conviction, not that I don't still enjoy working on classical Shannon theory things. I just felt that a great information problem, maybe the great information problem, is up inside here, and maybe some of us from information theory should start to try to look at it and see what we, if anything, we have to say. You need to be careful when you go into a quite different discipline, not to come marching in with your favorite hammer looking for the nails that it's going to knock down, because most everybody who does that in brain theory, coming in from some particular discipline, that they mistakenly think that the brain is a subset of their discipline, they soon find out that they couldn't be wronger. And I know very well that not information theory and system science alone are enough to solve this problem, but I think they can be used very, once you can convince enough people that theory could possibly matter here, then I think they can play a big role in helping to build that theory. So, okay. So we're trying to model and analyze certain biological phenomena, and that's both, I think, a good thing for information theory and a good thing for biology if you can do it right. So, this way is. So here's what I think is some, quote, good information theory that doubles as maybe good biology. One thing here is intra-organism information transmission via chemical processes. Maybe diffusion is too specific a word to use. But I won't probably talk about that in either of my two lectures beyond the little bit that I'm about to say right now. In living systems, information is the time variation in the concentrations of chemicals in compartments. Now listen, I said that sentence in a certain way. I didn't say that the information is represented by that, those concentrations. I said that it is that. That's what it is. And the time variation of how chemicals change their concentration in different parts of your body. The neurons are parts of it. The stomach and the kidneys are other parts of it. That stuff is information. And you can, well, but not today, not on this. I've done some work on efficient techniques for cDNA, library screening, monoclonal antibodies, and so forth. That's the stuff I did with Jim Mandel. Really not relevant to this morning's talk, but I think it's good information theory and good biology at the same time. There's something of a connection there to block designs into algebraic coding. At the end of this talk, I'll talk about an experiment of about 10 years ago now by Bill Balick and others that established Shannon mutual information rates between sensory stimuli and neural responses. And I consider that this was a major, major step forward in the theory. Balick is very good. He came into neuroscience from physics, but he's been working in neuroscience for probably 20 years now. And he has a good mathematical mind, understanding of a physicist, and he knows the biology. This is just one of many, many good things that he's been involved in. But it's probably worth saying a few words about what was done there. Prior to that article, I would say anything that tried to talk about the capacity of a neuron channel or capacity of the optic nerve, that was one of the favorites. Let's get the capacity of the optic nerve. And so what they would do is they would measure these spikes and other kinds of signals that are going up the optic nerve, and then they would do big statistical analysis to find the joint distribution over a large number of samples over a long period of time, some kind of quantization in the possible levels, and then they would compute what we would think of as some high-order approximation to this hopefully stationary random process's entropy. And this, they would say, is the capacity. But this has nothing to do with the capacity. Well, not nothing, but this is not the capacity. I mean, what are you measuring there? You could be measuring total noise. We don't get the capacity of a channel by measuring the data rate of the output. If there's a noisy channel and you haven't put anything in yet except the noise that's coming out the other end, and you measure that, this is not the capacity of the channel. The entropy of that is not the capacity of the channel. And these people didn't appreciate that, but Balick appreciated that. And what he did, and I'll explain it to you perhaps later on, was to set up an experiment in which he not only calculated the entropy of that output, but he also calculated the entropy of that output conditional on the stimulus that he was applying to the system somewhere else. And that was very tricky to do, but he did that so that he could subtract these things and get the mutual information. He could find out how much of the information that came out of this thing was the information was due to a particular stimulus. And now, of course, this has been extended, and people do this for, it's still pretty hard to do it for a very large number of places at once, but this thing's moving forward so that mutual informations for blocks of different measurements perhaps can be done. These experiments are very time-consuming. It took something on the order of 9 hours of recording to be able to make that one statement about one neuron and one fly. It's very difficult. But at least it has been approached the right way by people like him. And then there's perception of sensory information via neural nets with feedback, and that's really the only thing that I'm going to treat in any depth this morning. So first we have a multiple choice quiz. The inaugural Shannon lecture was delivered, of course, by Shannon, and it was devoted to one of the following 4 things: sources and channels, information measures, feedback, stock market. So let's see, A, B, C, D. How many think A? Nobody thinks A. How many think B? Nobody thinks B. How many think C? Well, a few people think C. How many think D? No, C's are right. Feedback. He did give a lecture on stock market. I went to his lecture on the stock market at MIT. It was scheduled in a room about twice the size of this one at 4 o'clock in the afternoon or some weekday. About an hour before, you couldn't even get near the room, let alone in it. They moved it to some giant auditorium where and even then there were other people who were pushed out into some things where they just heard the audio. He talked about it, and he had a theory of it that tied it to, he had made a lot of money in the stock market, and he did have a theory, some things to say about Brownian motion with drift and some things that you could do, and he had a neat little formula, compact, sort of like E is equal to MC squared type thing. The great formulas always have 1 letter on the left and an E and then 2 letters on the right with possible power on something. So he had one of those. But in the question period, I think it was more revealing when somebody asked him whether he had used this theory to generate his wealth in the stock market, and he said no. And then he was asked how he did. He said inside information, which at that point was not quite as illegal as it subsequently became. He was on the board of directors of Teledyne and so forth. Yes, feedback. So that's the answer. So here are some things we know about feedback. As information theorists, feedback sometimes increases capacity, sometimes doesn't. We do know that it, even if it doesn't increase capacity, it can certainly strengthen the performance in the sense that you can achieve rates of transmission through certain channels that have feedback with complexity of the encoding and decoding scheme, which is significantly reduced relative to what it would have had to be if you didn't have that feedback. Of course, it enhances stability. The control theorists know that. And it's a hot technique. We have still have a lot of stuff going on with Baum, Welch, hidden Markov chains, the BCJR algorithms for things like speech recognition, the turbo decoding, message passing, belief propagation, all that kind of stuff is some kinds of feedback, or feedback and feed forward and feedback again. So, and then there's Orlitsky's award-winning theory of sequential communication, where you talk and then I talk and then you talk and then I talk, and one of us is trying to get some message to the other. I had a joke about this in the Shannon lecture, but I won't take the time out today to do that. So you have that lecture has a compulsory joke. You have to tell at least one joke. But this morning I don't have to tell the jokes. That's why I put that there. All right, no, nobody's going in the wrong direction. So I would like to say that information theorists largely have restricted their attention to what I would call inter-organism communication. Organism here is being used in a very general sense that would possibly include things like computer-to-computer communication, or man-to-computer communication, or computer-to-man communication, and so on. So the quote organism, at least on this slide, is not necessarily alive. So here's some stuff about the nature of this inter-organism communication. Well, there's a source, and then there's a source encoder, and a channel encoder, channel decoder, source decoder, user. Figure 1, right? I once worked on a patent case where somebody patented figure 1. Not quite. The channel was a wireless communication channel. Other than that, patented figure 1. And the worst part is he got that claim through the patent office. Of course, it didn't stand up, but unbelievable. There's one other story associated with that Dave Slepian told me. We're walking on some Wednesday in one of the excursions on an information theory thing. I think it was the one in Ulm and in Germany. And we're on the same bus. He was talking to me, and he was telling me about the lawyers at Bell Labs' response to his work on group codes. So when Dave invented group codes for error correction detection, people kind of excited about that, and Bell was looking into patenting this. And one day, one of the lawyers came to Dave in his office, and he said, Dr. Slepian, this work that you've been doing on group codes is very interesting, and we're excited about this patenting possibilities. But do you think we could possibly get a patent on groups? Okay, so in here, the channel is given when we solve these problems. Maybe we have to analyze it stuff to understand how it works, but it's given in the sense that it's some underwater scattering mechanism, it's some kind of telephone line, it's some kind of portion of the HF spectrum the radio frequency spectrum that you can use for wireless communications. The government opens it up and it says, okay, you guys, you can have this piece of spectrum, but you can't put more than this much power into it, and you've got to keep the stuff that's outside the band down by many, many dB. And yeah, there are a couple of scientific instruments and other junk in here, and we can't shut them off. You'll just have to work around that. So that's the sense in which I mean that the channel is given, and then we go about trying to design the system effectively. And the channel behavior is independent of the source statistics. The channel sort of sits there, and whatever you stick in, the channel responds to it in the same way. And we know that to use it efficiently, usually that requires codes with long delay and complex encoding and or decoding operations. And the source and the user have to have a kind of a language that they exchange these coding rules a priori, so that when you send somebody an index, 3,807, they know what that means. Okay. So now I want to talk about intra-organism communication. of the kind that I'm more interested in this lecture, and try to contrast it in some ways with that. Inside living systems, channels are not given. They adapt their transition probabilities over eons or sometimes over milliseconds in response to the empirical distribution of the data that's coming in, not to mention the data that's fed back from elsewhere in the system. So, but the important point is that these channels aren't given to you. These channels have already been optimized, or very nearly so, for the problem that they're trying to solve. That's why you hear and why you see what you do, because for countless millennia, these things have been in the process of adapting themselves to a certain problem. They're built to solve a particular problem. And they know the statistics of that problem. And when the statistics change, they change. Sometimes very efficiently. Some of them are very good at adapting, and others are not so good at adapting. It is certainly not true that the way in which the channel behaves is independent of the signal that's coming into it. It's constantly adjusting itself to that. So the time-varying joint source channel coding somehow gets performed efficiently by biological systems that have the appropriate, what I would call it, chemotopology. That's the thing I was talking about before, about how to set up compartments and change the concentrations of chemicals as a function of time in the right way. This last sentence is kind of disturbing to the information theorist. It says that little, if any, coding occurs in the classical sense of the term in interorganism communication theory. What do I mean by that? Well, our favorite code, at least as far as the theory is concerned, are long block codes. I don't think there are any long block codes in biology, intraorganismally. The next slide will take care of DNA. Obviously, I need immediate reaction as well. What about DNA? But so that's the next slide. We'll talk about that. Wait, what about DNA? Wait, what about it? Ah, well, it didn't. I was not patient. Yeah, it's a long block code. It has a discrete alphabet. It's got a lot of redundancy in it. Maybe that redundancy is in there in part to control some kind of, let something patrol the DNA and prevent errors from infiltrating into it. Maybe. But the important point, an important point to realize here, is that DNA enables two organisms to communicate. It's not designed, in that sense, it does other things too, but in that important sense, it's designed for interorganism communication. Okay? And that's a totally different problem. And that's why it has to be like that. There indeed, the rules have been exchanged beforehand of how to interpret this cell. When this cell gets over here, this sperm gets to this egg, and so forth. Rules are set up. You know. It's not happening just within person. But of course, the DNA also controls the expression of genes, and that's very much an intraorganism process. And so, if you're going to build a comprehensive theory of intraorganism communication, somebody needs to address that. Well, a lot of people are trying to address that subject. It's a terribly difficult subject, and I'm certainly not going to get myself involved in it today, but I appreciate that that would need to be done. Again, it is a question of chemicals of appropriate density in the right places, and in this case also at the right times, or in the right sequence. Because when you turn on and turn off one of these protein factories on the gene, it happens because certain chemicals get to the right place at the right place and right time at the right concentration to snap this thing on or off. Terribly complicated, and I certainly don't understand the proteomics and so forth. But what I do want to say is that this joint channel source coding gets achieved without the use of any long block codes or even many of our other favorite things, convolutional codes. A little more reasonable if there could be something like a convolutional code, but I don't think there is. And turbo codes, LDPC codes, probably not LDPC codes, maybe some of the stuff that goes on in turbo coding with the belief propagation. I think that does happen biologically in a bunch of contexts. So that's maybe the closest that we've come through our own work. But what I want to say here is that robust channel optimal performance without coding can be achieved in certain kinds of problems. And here are two examples. The first one, well-known problem, you have a source and it puts out IID, normal, 0, sigma squared random variables in discrete time. And you're interested in recovering it someplace with small mean squared error. But that place is separated from you by an additive white Gaussian noise channel that you have to encode the source over and put it through the channel, decode it, and then feed it to the user. So that is a power constraint on the channel. Can't send more than S joules of power per use of energy per use of the channel. And the best performance you therefore could possibly get in terms of the distortion, the mean squared error that could be achieved, is of course obtained by equating the rate distortion function with the channel capacity. In other words, entering the rate distortion curve at the point where R is equal to C and seeing what D is there. Okay. That little less than 1 minute statement took 15 minutes in the Shannon lecture, because they're not talking to people who know anything about rate distortion, and half the audience doesn't even know anything about channel capacity. So, but today, you know, we can skip that right away. So you equate C to R of D, and then you solve, and you find that D is sigma squared over the inverse of 1 plus the signal to noise ratio. But you can get that minimum possible mean squared error per unit variance without doing any coding at all. You just take these things as they come along, scale them by square root of S over sigma so that you achieve the, you don't violate the power constraint, stick them directly into the channel, add the noise, put a Bayesian type of scaling thing on the other side to estimate the mean squared error, and it comes out right at that D. No coding, no decoding, just a scaling instantaneously. Yes?
But it's not clear you can do this for multiple sources, right?
What?
You cannot, it's not clear you could do this for multiple sources, right, even if they were correlated.
Well, I didn't do that here. I don't know. There are certain extensions of it that I know do work, but if you have a particular one that you're interested in, we could talk about that after. I mean, you can put some feedback around the channel, and you can do bandwidth expansion, compression, some things like that. But I don't know how generally. I'm not trying to say that. Well, I am later trying to say that this happens very generally, but not that necessarily that it happens in this example. Okay. So that somehow seems this. Well, you know, this is such a special example. Everything's Gaussian, everything's mean squared error. How can we possibly believe that anything that works, how can we extend anything that works for that to things more general? Well, for good information theorists, we know darn well that we better be damn careful not to assume that just because it works in some Gaussian mean squared error thing that it works robustly. But, yes, David?
So you start with the discrete time, right?
That was discrete time, yeah.
Bandwidth match between the source and the channel.
Yeah, the channel's matched to the source is matched to the channel. That's what's happening here. Yeah, this is in discrete time. You can certainly extend this particular thing to certain kinds of continuous time situations as well, but you couldn't do it instantaneously like that. If there's memory in the source, you'd have to do something different. So I guess if you say have an additive band limited Gaussian channel, I don't suppose you could do this. Maybe if you have a power limited, completely white Gaussian channel, maybe you could, I don't know. But this is definitely discrete time right now. Okay?
And not only discrete time, but also exactly the same rate. I guess the point. Maybe I'm not.
Once again?
But this is also assuming exactly right? So suppose the source is twice the bandwidth as the channel.
Yes.
Then every time you have two samples coming.
Yeah, then there's two of them.
You wouldn't do that. You couldn't.
No, then that wouldn't work either.
Simply,
right? There would be
so I'm
problems.
just wondering how robust this kind of.
Well, these are good questions, and I don't know the answers to them right offhand. I'm just trying to point out that there are some situations that do work, and of course they are sensitive to a whole bunch of things. They look to be perhaps very special. Well, that is a special example, no doubt. Okay, here's another one. Suppose that you have an equiprobable Bernoulli source, again putting out 1 letter a second, let's say, and you want to measure how well you do by Hamming distance or normalized, really error frequency and recovery of the data. And you have a binary symmetric channel over which you have to send it that has some crossover probability P. So C is 1 minus h of P, R of D is 1 minus h of D. And you equate the R of D to C. This time it comes out with D is equal to P. I'm assuming here that both D and P are less than a half. Otherwise you have to do some min and P, 1 minus P, and so forth. But obviously you can get that even without the scaling that you had in the other problem. You just take the data and put it into the channel, and it comes out wrong a fraction P of the time. And P is the least D you could ever get, no matter how big a long block code you did, and how complicated you built the encoder and decoder, you would never do better. Okay. Well, I pointed these things out some years ago, and Michael Gaspar and Bixio Romoldi and others wrote papers about extensions of that, so you can find a certain amount of how you can extend this so-called double matching idea with some robustness, but certainly I wouldn't want to say with complete robustness. Okay, but I do want to say some things about. Okay, in these two examples, you get Shannon optimality because you've got the source matched to the channel in the sense that the distribution of the source is the thing that is the distribution that achieves the capacity of the channel, and similarly you have the channel matched to the source in the sense that the transition probability assignment of the channel is the transition probability assignment that solves the rate distortion problem, and so you put these things together and it works. That's a double matching. Now, I contend that most biological systems have evolved to be doubly matched like this. And this, they handle the data optimally with minimal, if any, coding and negligible delay. But you've got to remember, of course, that the channel was not given. The channel was built with a purpose. So this could conceivably be. And I'm almost positive that it's true of the channel part. As for the source part, being matched to the channel, that's less clear. But I think the channel has been designed to be matched to the source. It has no other purpose in life but to send this source. And if the source changes, it has some adaptability to continue to do it well. So anyway, I say here that information theorists of late are beginning to appreciate the fact that I think living organisms have been capitalizing on for eons, which is that you can get what we would call near Shannon optimum performance in many situations by relatively simple probabilistic methods. And many of these things employ feedback either around a channel or in the decoder from one part of the decoder to another, belief passing, Bayesian belief propagations, and so forth. All very probabilistic. I'm sorry for those who are algebraically oriented. I don't think it's algebraic. I don't think anything about it is algebraic. But then I'm a Shannon theorist. You know. Maybe I don't want it to be algebraic because I don't know that stuff very well. You have a question?
So one of the reasons I asked about multiple sources is if you did encoding in the neural system, it's probably going to be a population of neurons, right?
Well, I'll try to talk about populations of neurons later in this talk, so maybe I'll say some things about them.
But
yeah.
do you suspect it's similar?
To some extent, yes. Yeah, to some extent. Actually, this talk will be directed much more at regions of the sensory system neurons. The afternoon talk, but more at a single neuron behavior. Okay. But there's more, though. Living organisms are energy aware. Footnote there says that the brain consumes 25 to 50% of the total metabolic energy of sedentary human beings. 50% for little kids and 25% for people our age. This is, do I believe this number? I don't know. Some person who wrote an article about it believed it. I haven't heard contradictions to it, but I don't know exactly how you really measure this, but it's a significant fraction. And therefore, if it's that big, you certainly don't want to waste that much of your energy if you can avoid it. And I think that these systems are optimally doubly matched over a wide range of power consumption levels. Okay, and they change as needed. Let me talk a little bit more about this energy idea. Here's a badly drawn picture of the C equals a half log 1 plus signal to noise ratio, Shannon additive white Gaussian noise channel capacity curve. And what I want you to note here is that the curve has a slope, and the slope is information which is being measured in bits per second versus this power consumption thing on the bottom, which is measured in joules per second, so that the slope is a ratio of those, and therefore it's equal to bits per joule. And the more information you try to send through in the sense of getting the capacity to be up high by increasing the power, the fewer bits per joule you get, because these curves are always convex this way. Right? So you've got to appreciate that if energy is very important to you, you may have to sacrifice the speed of transmission. Biological, this is a special kind of channel, of course, but I think this is kind of the shape of this curve is always true, and if you're power constrained in some way, then it's always going to be true that you're going to pay for that for higher data rate with worse level, with more joules per bit that you get through. Not only you put through more bits, but you'll pay more for each one of them in terms of energy. So you don't want to do that unless you have to do that. Biological systems are very smart, and they have more than one mode, and when they need to do something in a hurry, they do it in a hurry, and they expend a lot of energy. But most of the time, that's not the case. They work down, I believe, that they try to figure out how to solve their information theory problems without using up much energy. The whole long-term trade of how you stay alive and how you propagate to the next generation has to do with how you use the information that you gather to get more sources of energy so that you can continue. You know, this is, that's it. But yes, there's special things, adrenaline and all the rest of it, that are built in there that allow you to up the speed at the cost of bits per joule, well, joules per bit, let's say, when you need to do that, because they adapt to that as well. Now I want to talk about neurons. And here I do have to spend a good amount of time, because most of you are not very familiar with neurons. I won't even bother to take a poll. I say here that neurons are magnificent, and they are. This is a quote from somebody named Gregory Scott Hickok, UC Irvine. It all boils down to neurons and the connections among them. Everything you see, hear, and feel, every movement you make, all your thoughts, desires, and plans, even your emotions and personality, can be explained ultimately in terms of patterns of activity in networks of neurons. You have a certain view of the world that's completely a consequence of the fact that your neurons fire at certain ways at certain times and are connected with one another the way they are. Everything that you know, everything that you feel comes from that. When I look out over the audience, I see the audience, but I don't really see it exactly. Something happens in here that lets me see the audience.
Is that a philosophy or science?
You got, it's, this is, you know, this is a fact that you don't necessarily disagree with it when you see it, but somehow you live your life not thinking about it that way. But that's the way it is. Does mental experience transcend the activity of brain tissue? Maybe. But scientists at least to date, don't have anything that proves that there is a difference between mind and brain. It goes on, and well, it will probably go on for many more centuries as a raging controversy. But scientifically, there seems to be nothing that establishes that somehow there's more in the brain than just the science. There's more in to what you mean by your mind than just the science of what happens in your brain. Surely I will not address that today. But anyway, here's a lofty but perhaps attainable goal, which is to someday understand the infobiology of the brain. So I want to talk about neuron cardinality. There are about 10 to the 11th neurons in the human brain, and most of them are formed between the ages of -1/2 and 1. Each neuron forms synapses with between 10 and 10 to the 5th others directly. I mean, in direct connection of, and that's really actually often in most cases a in degree and an out degree. Each neuron is connected through its axon to, say, in primary cortex, 10 to the 4th others on the average. Not only that, but it receives at 10 to the 4th synapses on it similar signals from other cortical neurons. So the in degree and the out degree are enormous, but the connectivity is very sparse because there's 10 to the 11th altogether. So it's both sparse and at the same time it's very high degree. Okay. Each neuron, okay, from age -1/2 say about 3 months into pregnancy, up to about age 2, the number of synapses increases, get this, at a net rate of 1 million a second, awake and asleep. Okay. And many of these connections are also broken, not just made in there, but they're kind of somewhat plastic. You can make them and break them. Mainly you keep most of them and adjust their weights, but there are extreme adjustments of weights that constitute setting the weight down to 0, which means breaking the connection and then making new ones. So that goes on at a tremendously high activity. And it's kind of believed that it starts to decay rapidly, especially after you pass 2. But more and more, it seems that people come up with some indications. No, maybe it's past 6. And now, you do keep forming them for your whole life. But nonetheless, I think most of this activity is kind of over with by age 2. And after that, you're adjusting connections. Now, you have to understand that these are net numbers of neurons. It doesn't mean that you don't make new connections and you don't make new neurons. How do they get this? It's not like they actually look inside of somebody's brain and see that happen, but they have neuroanatomy studies of brains of people at different ages who died for some reason or other, and they look at the connections and they see how many there are, and they see how fast they change in number, and then they see that they stop changing. I mean, so this is sort of that doesn't tell you that you haven't gotten rid of lots of others and made many new ones. And certainly that you could have interconnected them very differently, and for sure you changed the weights. You're still doing that all the time. Right now, you're all changing, as I am, weights in your brain, even as you listen to this lecture. Okay, here's an artist's conception of a neuron. There's a cell body over there at the left. There's this whole dendritic tree, and on the whole dendritic tree are these, say, 10 to the 4th synapses, a few more also on the cell body. And then there's an axon, which often is myelinated. It depends on where you are, and there's a myelin sheath around it, perhaps, but not so much in the visual cortex. But things that have to travel a big distance get protected, sort of like being inside of a transmission line. This is not wireless. This is very wireline. And in order to connect to one place or another, you have to break that sheath, open it up, and bring the tissues into correct. So. And a neural impulse runs down this, when it gets generated, runs down this perhaps myelinated sheath to lots and goes to lots of other neurons, but the axon often making many branches, sometimes just, okay. All this stuff is very, very tightly packed together. These things are very small. They're small, I don't know, the micron measurements off the top of my head, but they're packed in with very huge density, and all this wiring somehow is in there with that huge density.
Question. Just because you mentioned it's all wireline, do we know that? Do we know that there isn't some kind of wireless electromagnetic propagation going on inside the brain that makes one neuron fire somewhere and then another one somewhere else because it's the electromagnetic wave propagating across past the wires?
Two-part answer. No. Other part, maybe. I've tried hard over the time I've been in this to get a definitive answer to any question. Everything has an exception. Okay, everything.
Is there an exception?
I guess my feeling would be no. That doesn't.
Yeah.
Brainwaves notwithstanding, I don't think that happens.
Huh.
But if you're a extrasensory perception guy, you're certainly not going to accept that statement, and I'm not here to know how to refute you.
Well, I don't mean over long distances. I mean the distances within your brain.
Yeah, within the brain. I think if you want to get a signal from one place to another, you send it from your neuron to the ones that you're connected to, and you hope that they send something related to that to the other spot that you hope you may get to. Except that you don't hope at all. You just do what you do. And what you're hoping for, it doesn't happen, because when your signal gets to the next neuron, it's only one of 9,999 others that are there. It's lost in the soup, and yet somehow it works.
Okay.
Yes.
Because you have so many connections, when you have any input,
Can you say a little louder? And it'll help other people even more than me. Yeah.
when there are so many connections, when some inputs are received, multiple inputs, they never get mixed up. There are separate connections for each one which never get mixed up.
Well, they each have their own place to go to on on the receiving neuron's dendritic tree, each one of these connections is in a different spot, so they don't get messed up in that sense. What does happen, of course, is that the neuron does not preserve all 10,000 of these inputs as a function of time. It combines them in a certain way. That's network coding. I'm going to get to that. Neurons know about network coding. They are huge on network coding. They knew about that long before Raymond Young and Bob Lee found their butterfly example. Raymond is a student of mine, you know, by the way. I'm tremendously proud of Raymond. He's far transcended me already. Okay. And then those are not the only things in the brain, these neurons. There are also these glia. There are about 10 times as many glial cells as there are neurons. And it's believed that glia, which come in several subclasses, astrocytes and oligodendrocytes and so forth, have as their purpose the care and feeding of neurons. This certainly is part of their purpose, but I would be shocked to find out that we don't ever find out that they're doing a heck of a lot more than that as well. We just don't know much about it yet. I don't know much about it. My son-in-law is a glial scientist. So if you want to find out more about glia, you put into the web browser glia.org and go from there. There are, again, many meetings of multiple people who study glia. But we won't. Now let's meet some real neurons, not just artist conceptions. So real neurons are uglier. So here's an electron micrograph of a neuron with some supporting neural glia that protect it. And I don't even know what is what in here. I guess it's little lines point to where the cell body is and certain kind of glial cells that are lying in there close to it, helping it get energy and fight off microbes and whatever else they do. Yes.
The magnification factor of this picture.
Excuse me? I didn't even hear where that was coming from. It was.
What is the magnification factor of this picture?
What is the
magnification factor?
Oh, for that.
How small is it?
Well, I guess it depends on this some magnification for how it's projected on the screen as well. So I don't know. I guess you're saying how small is that thing? It's probably a picture of micron or smaller sub-micron dimension. Yeah, I don't know. Does anybody here know the answer to that? Is that close to right? Yeah. Yeah. Okay. Here is a stained picture of the dendrites and the axon and the cell body of I think it's some kind of thing in a flying fish. I don't know that it's really that color, but they stained it that way. So they can look kind of pretty if you put them on a somewhat, if you back off about 2 orders of magnitude from the previous picture, then it starts to look nice. Maybe that's 3 orders of magnitude by now. Okay. These dendritic trees come in many, many different varieties and different species and in different parts of the same species. And these are pictures of dendritic trees of various types of neurons, A, B, C, D through M, and I have, if anybody cares, I have somewhere a description of what those were. Here's one of them. This is a retinal ganglion in a postnatal cat. Not too many of these things. A little thing in the middle, of course, is the cell body, and here's this whole dendritic tree that gives you kind of an idea of the relative magnitude of what gets devoted to dendrites and axons as opposed to the center, at least that way. Here's a very small one from the retina of a larval tiger salamander. All right. Okay. This one is drawn by hand, way back at the turn of the previous century, and this is a Purkinje cell, Kajal, through this thing. Purkinje cells are in your cerebellum and they help you keep your balance. And they have about more connections than anything else. Those are the ones that have 10 to the 5 instead of just 10 to the 4. And he studied them and drew this picture. All right, action potential. Well, here are the phases of an action potential. An action potential is a waveform that at a certain point gets generated when a certain threshold gets passed, and we have more to say about thresholds in a bit. And then it's a pulse of this sort that propagates down the axon of the neuron all the way out to synapses on, let's say, 10 to the 4th other neurons, where it brings the signal to them. Okay. All that happens very fast. There's equations for this thing. First, Hodgkin-Huxley equations that quite accurately describe why this thing looks like it does and what the nature of the axon is as a transmission line to preserve the shape of this and to propagate it at a rather constant speed. All of which are very important, by the way. Not so much the shape, but the speed is very important. There's this mention of refractoriness, to which I'll come back in the afternoon. There's a concept of, I just lost one of my two speaker wires, so give me a second to. There's a concept of refractoriness, which is a thing that describes the fact that after a neuron fires, it can't fire again for a while. The length of time that most neurons need to have go by before they can fire again is on the order of 2.5 to 5 milliseconds. 3 is probably a good number to pick. Some people think that there's an absolute refractory period, up to 3 milliseconds, let's say, where it just can't fire no matter how hard you bombard it. And then after that, it can fire. Other people say, well, you know, that threshold curve, really, it's always finite. It's just that it's so large for here toward the left-hand end there somewhere. This picture shows it around 4 milliseconds that you can't get above it in time. So it might as well say that it's infinite. And then it falls down in here. And there's not really an absolute little moment known to the microsecond as to when you could next fire. I think that's probably a more accurate description of the situation. Okay. In most cases, the spike travels to the end of its axon in less than 2 milliseconds. Sometimes it takes a little longer because some of these connections, you wanted to get something to someplace far in the brain that's far away. Yeah, you can do that. You can build a connection that goes far away. And that happens. You made a direct connection that goes far away. But these regions talk to one another, and they're not necessarily all that close together. So some of these things do run a long distance, and those might take twice that long, maybe 4 milliseconds after they were generated to get to some of the most distant of the things. But in cortex, they're all pretty close together, and within 2-3 milliseconds of the generation of a spike, everyone who's supposed to get it has gotten it already. Okay. So there's multicasting taking on here. Viewed as a communication network. Every time I step over here, this happens. Viewed as a communication network, this human brain simultaneously multicasts 10 to the 11th messages that have an average of 10 to the 4th recipients. Okay. It does that at a data rate, although they work asynchronously from one another, but if there were a clock, this would be happening every 2.5 milliseconds. So I said on this slide 2 milliseconds. Every 2 milliseconds, a new binary digit is delivered to these 10 to the 11th times 10 to the 4th, which is 10 to the 15 destinations. 2 milliseconds later, another petabit, word bit is dangerous here. Binary digit, which I used earlier, is better because there are many more zeros than ones. So if you're talking to a group of information theorists particularly, they know how to distinguish between a bit and a binary digit. So I'm really talking about binary digits. They don't each carry 1 bit of information. Many more zeros than ones. Non-spikes than spikes, okay, in those little 2 millisecond intervals. Anyway, 10 to the 15th of these things get generated and delivered. And 2 milliseconds later, another 10 to the 15 of them get sent out. And that one that gets sent out come from processing the previous multicast. Damn, got to be a different way to do this. I'll clip it up higher. Get it up on here. So what I like to say here on this particular, I'm just going to hold it here. What I like to say in this particular slide, the next thing, is that not only are we seeing network coding here at the highest possible level, but the total amount of stuff that goes around The internet pales in comparison to this. I'm not trying to say that the internet doesn't, by now maybe it pushes around this many binary digits per 2 milliseconds, maybe. But most of that has to do with store and forwarding, and it's not getting them to the destination where they are intended for, much less absorbing them there and calculating something else to send out within the next 2 milliseconds on the basis of what you just heard. So if you put all that together, you see that this, I don't know, put it on and see if you can find some way to make it stay on, but it hooks on the little thing because I'm not using the clicker. It hooks on there every time I go by to push the thing. Okay, so this is just this particular view of multicasting was set up, of course, to make the brain, you know, appear super powerful relative, for example, to the internet. So it has a certain bias in the way in which I did this, but I don't think it's altogether off base. It is not unfair to say any of the things I said, although I suppose other things could be said on behalf of the internet to make the argument differently. Here's a very simplified diagram of interconnections of regions in the visual cortex of primates. So these regions have names, and they're called things like V1. That's the visual region 1, and V2, and V3, and V4, and V5. I'd actually never heard anything about V3, but
I'm trying to just put the keyboard on top of that.
Put it up on there. Okay, and close that, and then it won't happen. Yeah, okay.
Just to make sure this comes out long enough. Yeah.
That'll do. Thank you. Okay, so and what happens here is that in addition to lots of internal connections within each of these regions that I didn't bother to show in this picture, so-called horizontal connections, there's a huge number of those. There's also things going up to V's with a higher index and things being fed back to V's with a lower index, and sometimes skipping over some of these connections, skipping over one of those regions. These regions are not just imaginary constructs that somebody thinks that, well, this thing must exist here, like the ego or the id. These things, we know exactly where they are and what, to a large extent, what functions they perform and how many neurons are in there and how they're interconnected. This is, okay, well, what does it mean to say that this thing is a region? Why can't you just pick out any, you know, let's say there's 50 million neurons in this thing. Why can't you just pick out any 50 million neurons and say that they're a region? Well, it has to do with the connectivity. If you just picked out a neuron, 2 neurons at random from the brain, and there were 10 to the 4th connections per neuron, there would be 10 to the 4th divided by 10 to the 11th or 10 to the minus 7 probability that they would be connected. In these regions, the density of connection is hundreds of times greater than that. So these things are working together very tightly on certain problems. This is well understood. But I love this part of the quote. It says, in addition to all of the connections from V1 and V2 to V3 and V4 and V5, each of these regions connects back to V1 and V2. These seemingly backward or re-entrant connections are not well understood. Information, instead of flowing in one direction, which was the view for many decades, even the view of people who won Nobel Prizes for it, now flows in both directions. After the word now, I put lots of question marks and exclamation points, because obviously it's not just now, it's always been going on. You know, I mean, now the writer of this thing and others know that it's going on, but it's always been going on. Thus, the later levels do not simply receive information and send it forward, but are an intimate two-way communication with other modules, feedback. The whole thing uses feedback, and everything in the brain and everything in all of biology uses feedback, locally, regionally, and globally. And nested feedback loops that are terribly complicated. And any analysis that you do that doesn't take that into account, you might as well not even bother doing on a network level. What does this say for artificial neural nets? Well, artificial neural nets has been proven by Vatnis-Chernyshkov's theorem that you don't need any feedback. You can solve any decision problem that you want by putting in even one layer of hidden neurons, and you can make this thing match any kind of reasonable decision theory surface in a big dimensional space. So why do you need feedback? You don't need feedback to solve the problem. You need feedback to solve the problem efficiently with low energy and maybe a little less equipment. artificial neural network people are beginning to look at feedback, but because of the power of that theorem, I think they took their eyes off it for a long time because they felt they didn't need to think about it. That's a mistake in the same way that it was a mistake in real neural networks. So I was going to define a coalition, I called it here, but it's like a region, and that's what I said here, that this is a group of neurons that are usually locally close together, and they have very high connectivity with one another as opposed to the 10 to the minus 7 brain-wide mean connectivity density. And the remainder of the synapses in which the axons of such a region or coalition go to is that they go up in that picture before to, in the main, the next higher level in this hierarchy of regions, or they go down to the one below. They're feed forward and feed back, or as the biologists like to call them, bottom up and top down connections. So they say top down instead of feedback and bottom up instead of feed forward. Real neural spikes occur in continuous time, but for many purposes you can model things by discrete time. The natural step to take is 2 milliseconds, kind of the width of one of these neural spikes that propagates 2 to 2.5 milliseconds. But I'm pretty much of the opinion now that that's inadequate, and you probably better split that down by at least a factor of 2, and more likely a factor of 5 to 10, because of high signal-to-noise ratio that allows for spike splitting upon reception. So here's a figure of neural information theory figure for a region of these things. Let me take my little laser thing. Okay. There are signals that come in, those are the X's, and some of them come in from above, they're top down, and other X's come in from below. These are lines along which spikes and non-spikes occur every couple of milliseconds. And as one of these lines comes in from some axon in the top, in a region above and top down back over to here, it doesn't connect to all of the guys in this region. I'm trying to say here that there's one line in here for each of the large number of things, neurons that are in this region, which tends to be tens of millions. But this thing connects, you know, much more intensely than brain-wide average, but not to all of these. You know, may connect to 1 out of 100 of them as it comes through, and another one that connects to a different set of 1 out of 100 of them, and so on. And that all comes down. And other things are coming up from more from the sensors down below, or similarly coming in from the region below and connecting in some pattern to some of the neuron dendrite tree and body, which is what these up and down lines are representing. And all that stuff comes in and it forms a postsynaptic potential. I'm not going to go into how that happens, except I would say that a very good model of it is that it is a filtered Poisson process. It's a really good model. One of our favorite things from system theory, and it really applies here. Anyway, these things get combined and they form a postsynaptic potential, and when that postsynaptic potential gets across that threshold that we talked about before, a spike goes out. And when it goes out, that spike, this is, I say, Y of k, is lots of spikes because there's lots of neurons. It's really thresholding of all these PSPs. There's many tens of millions of them here. This is happening. Some of them are going above their threshold at this moment and putting out a spike, and the other ones are just putting out 0 on their line. And that stuff comes out of here, all these zeros and ones, and it goes back across the region to talk to where it reconnects to some of the ones that are in the same group, and then it either runs up to the higher stuff or some of them go across and then go down to the lower stuff. That's sort of how it looks. Here's a fun picture of it that, oh, did I take that out? I guess I did. Let's see. Oh, that's why I put this in here. This is a marvelous experiment, in my opinion, one of many, but this one's really pretty, in which people demonstrated in a most convincing way the existence of this feedback in primary visual cortex. Primary means these first 3 or 4 levels of what's coming up. They found a neuron in primary neural thing. I think it was a V1 neuron. And this V1 neuron seemed to be involved in detecting, it responds to, in the sense that its firing rate goes up when the pattern that you're looking at has a vertical change in intensity. That's really in the horizontal direction. Along a short vertical line in a horizontal direction, there's a change in intensity in the pattern. When that happens, this one tends to fire a lot. So that may happen along a little bar like this, or on the edge of a picture, or a black and white thing that changes. And so I'm kind of talking about a neuron who in this picture is, say, somewhere along this line between here and here, where in fact there isn't anything. Okay, there's not change. And yet, depends on how you are, but most of you, when you look at it, will see this white square in the middle that comes out. You see it bounce out from the picture, from the white in the background, follows the little Pac-Man in the corner and fills in the line. It's black, but it's white, and a different weight in the background, but it isn't different, really. It just looks different. But it's exactly the same as the rest of the background. In other words, this popped out square is not popped out, really. That's just the way it looks to your visual system. This is a so-called Canizza square, or illusory contour. And what they measure here from this neuron is what happens when you put the line on that picture, or this picture, or this picture, or that picture. And you flash it on in the visual field of some monkey, in this case, whose head is being kept, and they're recording from something inside there. All right, and what is this picture for the case that you have the, I don't know, this dark line here that has to do with this little line, the bar. Well, what's going on there is time in milliseconds out to the right. And what they really do is they do this thing many, many hundreds of times. And then they tell you for each one of these little points in the histogram, it's sort of like a histogram of where the spikes were. So where they were when you had lots of them at this time in this little histogram bin, out of the 10,000 times or whatever number of hundreds of times it was that you did this experiment, when you had a lot and when you had a little. So here at the beginning you didn't have anything for the first 50 milliseconds after you flashed it on, and then it started to rise very quickly, and then it followed some sort of a pattern that looked like that, averaged over a very large number of experiments and put together. And when you flash the other patterns, you get different things, although the beginning looks the same, except this one. That's the illusory contour. When they flash that one on, nothing happens for almost 100 milliseconds. That's because the black line, the change in intensity, is not there. Okay? And so that neuron doesn't pick it up in V1. But after close to 100 milliseconds, it starts to do, in a weaker way, the sort of thing it did before. It ramps up and then it dies out quickly. Why does it do that? Well, the reason why it does that is that stuff has gone up by that time to higher levels. And somebody's looking at this picture and he says, you guys over here, of whom this one they're recording from is one of them, you show me that there's this line segment, and I know that there's this line segment, but you guys over here are responsible for that part. You're not telling me that it's there, but I'm trying to make sense out of this picture, and it could be there. If it were there, there's a reason to believe from the pictures I've seen in the past that that line might continue. Maybe something's happened. Maybe it's gotten covered up by sand. You got to know it could be a scorpion lying under the sand and only seeing part of the thing. I want to know whether that's there or not. So what do? They feed back some things that, in essence, one way of saying is they lower the threshold. They feed input that isn't coming from the sensory organs, but is coming from stuff higher up, to try to make it possible to see whether or not, with a little bit of help, you can see this thing, because it's faint for some reason. And eventually, they push hard enough that, in fact, they make you see it on the basis of what they sent back, even though it isn't there at all. Yes?
See, so the upper, the higher levels of the visual cortex figured it out. If they figured out, why do they need to give feedback for the lower levels, so for the V1 neurons
well,
to fire?
What I said, there was a possible explanation of what happened here. It does not really prove anything. But I don't know about in this experiment, but in lots of others, it's been more dramatic than that. What they've done is they don't necessarily know where these signals are coming from, but they find things along the pathway in higher regions. They cool them off. They cool those neurons, and it disappears completely, that red response. Flat, nothing. Let them warm up again. They're in higher regions, and it comes back. So there's no doubt that there's feedback paths that are responsible for this delayed response. And it's happening in an autonomic way in very low level before you're really even getting to the point of trying to decide whether this is a scorpion or a bear or nothing. All that feedback is very quite local and happening and controlling things like this. And why it pops out at your vision afterwards, I don't know. That's another thing that people study, but I'm more interested in the feedback aspect of it. That's a very nice experiment about that. Here's that same picture I had before imposed on a head, just to play around. And I like to then try to put some mathematics behind this and talk about what this regional or coalition channel might look like. So that, let's say, if one of these y's is a vector here, as time goes by, you get a vector at time 1, time 2, time 3. These times are separated by 2.5 milliseconds. Maybe finer if you want to sample finer, but something on that millisecond type region separation. And the x's are the stuff that's coming in from below and from above. And the y's are the stuff that are going out, which connect locally and then go down or up. All right. And these are big multi-million component vectors of zeros and ones, each of these y vectors. And we have one at each time instant. And you want to know how this thing behaves. And to a certain extent, it behaves that the joint distribution of the vectors of a succession of vectors of y's, given the initial y vector and the x's that came in over those times, is a product distribution in the sense that each component of y at time k either is a 1 because you produced a spike during that time, or it's a 0, and that depends really only on what is being imposed onto the neuron at that time, which is the new input at that time, plus the stuff that got fed back from the previous time. Now, there is not really a previous time in the real neural network because it works asynchronously. But if you take this time separation and take each spike that occurs and move it to the center of the nearest discrete time instant, you won't change things too much, especially if you get that down to be less than a millisecond. So think about it that somehow when you do this analysis, it takes one time step for the stuff to get fed back to show up. But the things that got fed back before are gone. That's kind of the problem with taking the time step smaller than the width of the spike, because they wouldn't be gone yet. But if you take it at the width of the spike, it's pretty much true that the other stuff is gone from last time. It's no longer affecting what's going to get produced next time. So this slide shows a neural correlation in this sense as a linear but thresholded random dynamical system. I'm not going to go into this equation, but this has a good amount of stuff about weights of synapses and quantal synaptic failures and things like that. But you can write down something that describes that, but I won't. What I'd rather do about here is to talk about a maximum information rate hypothesis. This is a hypothesis at the moment, that the input, or so-called afferent process that's bringing signals into this coalition, this vector of x of k's, will have the property that it maximizes the mutual information rate between itself and what comes out. Given it does that maximization over all the processes that lead to the same or smaller amount of expenditure of energy in these neurons that are producing the y vectors. So the x stuff is coming in from outside. Here's this collection of neurons, and they produce a bunch of signals, ones and zeros, on each of their many tens of millions of components, and every couple of milliseconds. And what I'm saying here is that this thing is worked out in such a way, has been built in such a way, this is a conjecture, that the mutual information between successions of vectors that come in like this and vectors that get produced, in other words, the amount of information that this thing sends out in response to what came into it is as great as it could possibly be given the energy that it used to make it. I can't prove that conjecture, but I can tell you very many nice things that arise from it if you make it. This is a certain kind of revised version of what neuroscientists call infomax. The original infomax thing is not phrased in quite the way that I think an information theorist would like it, but it had the same spirit. It's just that it didn't take the energy into account to the extent that it should have, I think. Here I talk a bit about how the energy is, what expends the energy, but I'll be saying more about that in the second lecture, so I won't spend time on it right now. And here are some justifications for why that would be. One of them is, again, this fact that the brain consumes 25 to 50% of the rest energy, and it would seem to be wise to make its computations be energy aware, energy efficient, so that you would want to get information without expending much energy to push it around to where it has to go. And it just seems inefficient to build and maintain such a network and then expend more energy than necessary to transfer whatever information about its afferent or arriving process that it conveys to the efferent ones, the ones that it talks to. It just seems that you don't want to, once you've got that thing built in a certain way, it would seem strange to spend more energy to send it the same information than some technique that could do it with less energy, so it should be done with the least energy. The maximum information hypothesis, and this is nice, leads to Markovianness of the joint input-output process, and that reduces latency by encapsulating the memory so that you don't have to keep remembering all the past inputs in order to get a complete statistical description. And that's leads to the theorem, main theorem really, that if the maximum information rate hypothesis is true, then this process of vector process of x's at time k and vector y's at time k put together over k as a time process is a first order non-homogeneous Markov chain. Sometimes it's easy to see what has to be done to make it homogeneous, which might be sometimes. The output process from the neuron group is also a first order Markov group chain. The input process is not necessarily Markovian. All this comes out of the math. And I'll give you, I guess not in this lecture because it's going to end in about 5 minutes, proof of this theorem. This is joint work with Yu Zhengying, one of my famous former PhD students who now works for Intel and doesn't do anything like this.